Hello, bio students. Today we're going to continue with the unit on molecular biology, and we are going to go over lesson 2, which is called DNA structure. So you're going to need the note taker that is for lesson number 2, and you're going to use the video to take your notes and follow along. So let's continue. Okay. So it's important to remember this whole central idea in biology, which is referred to as the central dogma of biology. And what does this mean? This means that DNA is the one responsible to carry the whole genetic information. But DNA makes RNA, which is kind of like its messenger, to then take the genetic information to ribosomes and make proteins. And those processes all have a name. So, for example, DNA can make copies of itself, and that's known as DNA replication. DNA can make RNA, and that's known as transcription. And RNA can make proteins, and that's known as translation. And we're going to remember these processes later. So this is the central idea or belief, which is referred to as a dogma of biology. So it's always important to keep this idea in the back of your head as we continue studying molecular biology. So let's start with filling out the chart that you have in front of you in your note taker to fill in the characteristics of DNA and RNA. So here we see a picture, and let me get my laser pointer. So here we see a picture of DNA, and notice that DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. Notice that both of them contain sugars. The difference is that DNA contains what is known as a deoxyribose sugar. It is a pentose sugar. That means that it's a molecule with 5 carbons. And RNA has what is known as a ribose sugar. So the sugar component varies a little bit. Both DNA and RNA have nitrogen bases, but the types of nitrogen bases that they have vary. So the 4 nitrogen bases found in DNA are adenine, cytosine, guanine, and thymine. And the 4 nitrogen bases found in RNA are adenine, cytosine, guanine, and uracil. So notice how there is no T, there is no thymine in RNA. So the big difference is that RNA contains uracil as that 4th base, and DNA contains thymine as that 4th base. As far as the number of strands, DNA is double-stranded and RNA is single-stranded. And remember, you can always pause this video if you feel that I'm going too fast and you need to go back and write down your notes. The shape, well, DNA is a double helix and RNA is just a single strand. Now, RNA comes in different shapes because there's 3 different types of RNA, mRNA, rRNA, and tRNA. But we're going to learn more about the shapes of those 3 types of RNAs later. So right now you can just say, as far as shape is, it varies. As far as the function of DNA, the function is to contain the genetic code for building proteins, and the function of RNA is to make copies of proteins. And then as far as the location, DNA is found inside the nucleus. It is too big to leave the nucleus. RNA is also made in the nucleus, but it can leave the nucleus. It can leave and travel through the cytoplasm to the ribosome. Okay. So let's look at the shape of DNA. Now, the first important vocab word that we need to understand here is the term nucleotide. So the nucleotide is the monomer or the building block of DNA. And we're going to look in more detail the actual makeup of the nucleotide in just a moment. But notice that DNA has two strands that are anti-parallel, meaning that they run in opposite direction. Here you can see how the sugar is pointing up and how the sugar on the complementary strand is pointing down. So that's how you know that they're anti-parallel, running in opposite directions. And then we see that the backbone is the exterior of the DNA, and the backbone is the sugar phosphate backbone. So sugar phosphate, sugar phosphate, sugar phosphate. Okay. Now the backbone is really strong. You cannot have the backbone of DNA fall apart. It would be disastrous. So the sugar and the phosphates are held together by very strong bonds known as covalent bonds. Now in the interior of DNA, we have the nitrogen bases that pair up. So here we see that adenine, A, pairs up with thymine, and guanine, G, pairs up with cytosine. So these complementary base pairs are always going to be the same. This is Chargaff's rule. And we see that the bases are held together by weaker bonds called hydrogen bonds. So when we study DNA replication, you're going to see how the bond between the bases can actually break so that DNA can actually make copies of itself. So in your note taker, it asks, what is meant by DNA being anti-parallel? So we know that the sugars are pointing in the opposite direction, right? So that's what it's meant to be anti-parallel. So let me skip a couple slides ahead here, and let me show you what determines that, that anti-parallel structure. So the carbons in the sugar, these pentose sugars, they are numbered. And we start our numbering after the oxygen found in the deoxyribose. So every time you see a little peak here in the pentose sugar, that represents a carbon. Okay, so this would be carbon 1 right here that I'm pointing to. This would be carbon 2, this would be carbon 3, this would be carbon 4, and then the fifth carbon lies up here. Okay, so right here we see a formation running from 5 carbon, and if you go down to a 3 carbon. So we call this strand a 5' to 3' direction because it starts with a 5' carbon and it ends with a 3' carbon. But if we look on this other side and we see that the sugars are upside down now the numbering stays the same, right? We start numbering our carbons after the oxygen, but now we see that this is carbon 1, carbon 2, and this is carbon 3. So we see that the direction now is running from 3', and if you move down here, you see that it's running in the 5' direction. So that's what it's meant to be anti-parallel, right? One strand runs in a 3' to 5' direction. The other strand runs in a 5' to 3' direction. And this is due because of how the carbons are numbered in the sugar. Okay. Okay, so let's go back to our description of the nucleotide, which we said was the building block or the monomer of DNA. So if we put many of these monomers together, we get our DNA structure. So the nucleotide is actually made up of 3 parts. So here we see the pentose sugar, that's one part. We see the nitrogen base, that's the second part. Now, again, the nitrogen base could be A, T, G, or C. And then we see the phosphate group, which is the third part of the nucleotide. So if we go back to our DNA structure here, we see that, let me draw this for you so you can actually see it. Here we see one nucleotide right here. Oops. Here we see one nucleotide, right? And here we see another nucleotide. And here we see another nucleotide, okay? So here we see the monomers or the nucleotides forming the DNA structure. And that's what it's meant by DNA being a monomer of a monomer of nucleotides. Okay, so moving on here. So let's talk a little bit about the bonds of DNA, and we've already seen that before. This is the second page of your note taker. So the bases, G and C, are held together by hydrogen bonds. And so the location of the hydrogen bonds is between the bases. Notice that guanine and cytosine form a triple hydrogen bond. And notice how adenine and thymine form a double hydrogen bond. And then what about the location of the covalent bond? Well, I told you before how the covalent bonds keep the sugar phosphate backbone very strong so that it doesn't break apart. So the covalent bond is found on the backbone of DNA, on the sugar phosphate backbone. Okay. So looking at the nitrogen bases alone, we can classify the nitrogen bases as being purines or pyrimidines. So what does that mean? Well, purines are molecules that look molecularly different than pyrimidines, and you don't need to know the whole molecular difference. All you need to know is that adenine and guanine are purines and that they are bigger in size. Okay, so you can see how much bigger the purines are than the pyrimidines. And then the pyrimidines, which are smaller, these are the cytosine, thymine, and uracil in RNA. So the way I like to remember who the pyrimidines are is by using this mnemonic, cut the pie. Okay, so cut stands for the cytosine, the uracil, and the thymine, and the pi for pyrimidine. It's just a little easy way for me to remember it. All right, so now let's solve, before we do that, let's go back and look at the purines pairing up with the pyrimidines that we now recognize as Chargaff's rule. Okay, G pairs up with C in a triple hydrogen bond, and A pairs up with T in a double hydrogen bond. So we can also say that purines, G and A, pair up with the pyrimidines, C and T. Okay, that's also what we recognize as Chargaff's rule. All right, so knowing a little bit about Chargaff's rule and the pairing of complementary bases, let's solve this problem. So if a DNA molecule contains 40% adenine molecules, how much guanine will be present in the molecule, and then how much thymine will be present in the molecule? So let's work out this problem. So we know that the amount of adenine has to be equal to the amount of thymine, right? Because that's Chargaff's rule. So here they're telling you that there's 40% adenine. So we know that if there's 40% adenine, then there's got to be how much thymine? Well, there's got to be 40% thymine as well. Okay. So together, the amount of adenine and the amount of thymine is going to equal 80% of that DNA molecule. Okay? So then that means that the remainder, the G and the C, is going to be equivalent to 20%, right? Well, how do we know that? Because 80 plus 20 equals 100%. So then the remaining part of the molecule, which is made up of the remaining 20%, has to be a combination of guanine and cytosine. And we know that the amount of guanine is equivalent to the amount of cytosine. So that means that the amount of guanine and cytosine need to be split up. So 10% of guanine is equal to 10% of cytosine, and that is going to be equivalent to the 20% that's left. Okay? So to answer the question in this problem, how much guanine will be present? Well, there will be 10% guanine. And how much thymine will be present? There will be 40% thymine. Did you get it right? Good. All right, moving on. So describe the forms of DNA packaging. So DNA looks different, and it depends on how you're looking at the DNA. So DNA can be packaged into different forms. Now remember that depending on the way DNA is packaged will determine whether or not you can actually look at the DNA. So when we extract DNA from strawberries, we're going to be able to extract a lot of the chromatin inside the DNA, and we're actually going to be able to see it. We're actually going to be able to see all that goo, right, that looks like snot. So chromatin in the nucleus of a eukaryotic cell, it looks like very loose DNA. Okay. Now, if you were to zoom in inside that chromatin, we would be able to see a more tightly wound up DNA. So we see here that chromatin, if you want to keep winding it up, think of a piece of rubber band that you keep twisting. So if you want to take this loose chromatin and you want to keep winding it up and getting it more tightly packed, more condensed, you're going to take that chromatin and you're going to wind it up around these proteins. And you're going to get a nucleosome formation. So a nucleosome is tightly wrapped chromatin that's wrapped around histone proteins. And if you keep twisting it, and if you kind of think of that rubber band that you keep twisting and twisting and twisting, and you zoom in, you're able to see the DNA in helical form, or known as a helix, which is your molecular double-stranded helical DNA. So of course, you're not going to be able to see the helix with the naked eye. You're not going to be able to see a nucleosome with the naked eye. Okay. Chromatin, perhaps you can see it with the naked eye if you get a lot of chromatin from different cells all bunched up. Okay. And then under metaphase of mitosis, you might be able to see this chromatin kind of assume a chromosome shape. So chromatin can condense itself into a chromosome. So chromosome is condensed chromatin. So if you're looking at how the cell is about to divide during metaphase of mitosis, you might be able to see some chromosomes under the microscope. And these are visible under a microscope. For example, if you remember in class, we've seen chromosomes of onion root cells. So here we see how the chromatin can be packed into a chromosome, and it assumes this X shape. And during our genetics unit, we saw the different structure within the chromosome, like the centromere, which is the central region of the chromosome holding the two chromatids together, which are the arms of the chromosome. Okay, so these are just different ways DNA can be packaged. So that's it for your notes, and that's it for lesson 2.